Welcome to our fifth and final lecture for our Chapter 3 genetic unit. This will be the last lecture for genetics as an intro. There is a secondary unit on genetics that's a little bit more advanced that we'll get into later. But today is all about genetic modification and biotechnology. So an intro to a bunch of cool new tools and things that scientists are doing in relation to genetics in research labs. So the first thing we're going to talk about is PCR. And PCR is an artificial method of replicating DNA. So basically what you do is you take a small initial sample of DNA and you heat it up to separate it into its two single strands. And then you have primers in the liquid that the DNA sample is in, and so those DNA primers attach to the target sequence, and then you elongate that. So then you have a heat-tolerant DNA polymerase called Taq, and that binds to the primers that are in the mix already, and it copies the strand, and it continues to copy and copy and copy forever and ever until you reach the amount that you're looking for, and so it will go on for a couple of hours and it will just synthesize new DNA so that you have a larger sample so that you can really look at it. For gel electrophoresis, this is a technique used to separate and isolate proteins or fragments of DNA on a larger scale. So the larger the DNA fragment, the less it's going to move on the gel. And so the smaller the DNA fragment, the lighter it is, right? Because there's less nucleotides in it, so it can move further through the gel. So it gives you an idea of the sizes of the fragments that you're looking at. For DNA profiling, this is a technique used to identify people because people have unique DNA. And so there are certain areas in your DNA, in non-coding DNA, where you have short tandem repeats, which is basically like 4 or 3 nucleotide bases in a row that just repeat, anywhere from like 4 to like 25 times, and everyone has different amounts of that, and so you can measure that and then use that to profile individuals to see if they match for paternity tests or for forensic investigations. This is how you can determine if the blood matches your suspect, etc., stuff like that. For gene transfers, this is when you are literally transferring genes between species. So this is gene modification. And then whatever you make and you insert that new gene into a new organism, that new organism is transgenic. So if I wanted to take a human insulin gene, but human cells replicate so infrequently, I could put that insulin gene into a bacteria plasmid, and that bacteria plasmid would replicate tons and tons and way faster. So I have that transgenic bacteria, right? I have that different gene in this organism, so it's transgenic now. I can grow it in culture and grow tons of it, but then I can extract that human insulin gene, and now I just have a ton of insulin that I can use for people that have diabetes. So the steps to this is to isolate the gene by using PCR, digest with restriction enzymes so that you are getting the specific gene of interest that you want, ligation of the vector, so you put the vector and the gene together with DNA ligase, so you make this recombinant, and then you can insert it into whatever organism you're using to replicate, and then it's into its new host, and you can replicate ad nauseum, really. For vector deliveries, this is how you would get that gene back into your original host. So there's non-viral delivery, in which you use a bacterial plasmid, and so there's 5 different ways that you can do that. You can use electroporation, heat shocking, particle bombardment, microinjection, or liposuction, and you can see what all of those do on the screen. You don't need to know the ins and outs of how they do that, but it's good to know that like electroporation uses electricity, right? Electro, and then pore is making holes in the cell membrane. So the electric current makes holes. Electroporation, I mean, it's named appropriately. So that's non-viral delivery, and then viral delivery uses a virus, right? So the virus integrates their DNA directly into the host genome. Of course, this has risks because this random insertion could change the host genes because the virus could take over. So there are some, pros and cons to both. One thing that we're going to talk a lot about is GMOs, and that is like the big thing with biotechnology. This is like a huge discussion that's still going on. And so I'm not going to go over the benefits and risks, but you should maybe pause and just like read this or print this picture out so you have it in your notes, screenshot this, put it in your notes, something like that so that you have it. It's really good, and there's like always a question about the pros and cons of GMOs and what you should know and what you what are some risks and how does the population feel about it, and where are we heading? So just all of that is like your general benefits and risks, pros and cons. So good to just have this in the back of your mind if you are unfamiliar with GMOs and their benefits. So some types of GMOs, again, I'm not going to go over all of these, but good for you to be able to differentiate them or have examples to pull from if you were to have a question about this. So GMOs, if you don't know, it's genetically modified organisms. So golden rice has been modified with daffodils, flavor saver tomatoes, had genes removed. BT corn had a bacterial insecticide gene added to it. Aqua Advantage salmon had a growth hormone injected, and then glow-in-the-dark animals have fluorescent proteins in them. And that one has no practical purpose, so that's a very much like, well, then why did you do it type thing, and that's when you have to have that morality question, like, is the science beyond where we are ethically, and if so, how do we bring it back to where we are in a moral standpoint so that we're not taking advantage of maybe organisms that don't need to glow in the dark, right? So in an example, GMOs and monarch butterflies. So that BT corn that I was talking about has that insecticide gene. So that means that it produces this lethal insecticide to the European corn borer, which is, the larva of the European corn borer. So then, from there were concerns about, well, how are monarch butterflies going to be affected by this? Because, they need to eat things too, and if you're adding this insecticide to things, are the butterflies going to be affected? And so they did a preliminary study to test the survival rates, and the caterpillars that were exposed to this ate less, grew slower, and had a higher mortality rate. So some suggested that this study was invalid because it didn't accurately represent natural conditions, and then so when that was brought up, they did a second study, and they found no significant risk to monarch butterfly populations. So, this also begs the question of, like, is all the information that is being done, is it provided to the general population? Because the preliminary study found that there was a definite risk to the monarch butterflies, but then some people said that that wasn't natural, so they did a secondary study, and then there was no significant risk. So why were the two studies so very different? Well, I don't know, and that's a question that a lot of people are asking, like, does the general population have access to all this information to make informed decisions? So moving out of GMOs and into clones. So clones are genetically identical organisms derived from a single original parent cell. So asexual organisms will always produce clones, but there's also conditions where sexually reproducing organisms can produce clones, like twins, right? Those identical twins are clones. So if you want to clone a multicellular organism, it requires stem cells. And cloned embryos can be produced by somatic cell nuclear transfer. And we're going to get into both of those, but I just kind of wanted to show you this picture, right? So you have skin cells that you can grab and destroy the nucleus of that unfertilized egg and transfer the nucleus into an egg cell and then grow the egg cell, right? Fertilize that and you get a cloned organism. So, you can make clones in a lot of different ways. So this is an asexual way, right? Binary fission, where you just kind of like grow and split. Budding is when you have like an extra growth that eventually splits off and you have identical DNA. And then fragmentation, which is like in starfish where the arm will break off and regrow. So the original starfish will regrow its arm and the arm can grow into a secondary starfish. Other cloning methods, like in plants, you have vegetative propagation where you take small pieces of plants and then grow them independently. And then you have spores, like algae, moss, and ferns, which can reproduce asexually. And then you have human cloning, where you have identical twins or non-identical twins, right? So you have one sperm and egg that splits into two and you get a shared placenta. Or you have two eggs, two sperm, and you have genetically distinct, right? So this would be like fraternal twins and you have separate placentas and they're grown at the same time, but they have different egg and different sperm. There's also artificial cloning, which is when animals can be cloned at the embryo stage by breaking up the embryo into more than one group of cells, right? It's normally one group of cells that grows into an embryo, but you can separate them out and then place them into surrogates, and then they will grow as if they were embryo. So they'll continue to grow, and then you will have identical offspring, which are your clones. And you can clone adult organisms. This is a more reliable method of artificial cloning. It involves somatic cell nuclear transfer. So you can do reproductive cloning or therapeutic cloning. Reproductive cloning is when an embryo is implanted in the uterus of a surrogate and a new cloned organism will develop. So you take that embryo and you just put it into a surrogate. And therapeutic cloning is when you take embryonic cells and you induce them to differentiate to create specific tissues or organs. And then those, once you purposefully differentiate those cells, you can take those tissues or organs and transplant them into wherever they are needed. So you can make red blood cells or nerve cells or liver cells or whatever you may need. Stem cuttings are used with plants so you can separate and regrow them to make a new independent clone. So you take a little piece of a stem and you put it in water and you let it regrow, and it's going to create new roots and new leaves, and it'll create a secondary plant. Of course, there's always factors that affect that, like where you cut it, how big it is, what you're growing it in, are you using growth hormones, and then the normal ones like temperature, is there water available, pH, and light. Now you can do all of this, and now we're getting back into like ideas of biotechnology, so screening for genetic diseases. And you can do this in two main ways, using cDNA and a microarray. So cDNA is complementary DNA, and it's synthesized from an mRNA template using reverse transcription. And then you would use that cDNA because it represents gene sequences that are transcribed by a cell. And you can determine expression patterns in normal and disease states. And on the next slide, we're going to go into a little bit more of that. And then a microarray is a collection of really small DNA sequences called oligos attached to a solid surface. And if the gene is active in a cell, the cDNA will bind to its complementary oligo, right? If it's active, it will bind. And if the cDNA has been fluorescently labeled, then you can very clearly see that cDNA because it's going to light up. And then with this, you can determine healthy and diseased cells because they're going to light up differently. So you can make nice comparisons. Now here's that example of this. So at the top you see a normal cell and a cancer cell, and it's going to go through the same process, and it's going to get reverse transcribed and labeled, and the normal cell is going to be tagged with green fluorescence, and the cancer cell is going to be tagged with red fluorescence. And as it goes through onto the microarray and then scanned, you can now see if it's significant, it's going to show up green in normal cells. And if it's in disease cells, then it's going to show up red. And now you can clearly see what's being affected and where. And then you can focus your work on these disease cells that have been flagged. So gene therapy is another biotech way to help with disorders and diseases. And this is when you insert genes into an individual's cells to treat hereditary disease. So you can harvest cells from a patient and then mess with the DNA in it and insert a new gene and mix that new gene with the patient's original cells. Now, of course, that cell then becomes transgenic, but then you insert the altered cells into the patient, and then the altered cells start to produce whatever protein it is that you're looking for. And sometimes this can work and become the new cell in your body, and your body will start making the protein that you couldn't make before, which obviously helps you deal with whatever the disease or disorder that you are working with before. It makes it a little bit easier for your body to fight off or help cope with it. For gene silencing, this is the ability to kind of prevent the expression of genes. So there's conditional knockouts and RNA interference. Conditional knockouts is a genetic technique where a specific gene is just rendered inoperative. So basically, I am choosing that this gene gets knocked out and it doesn't do anything anymore. And then RNA interference is short interfering RNA, which messes with the expression of genes. And it does this by causing the transcripts to be broken before you go to translation. So you can transcribe mRNA, but you kind of like interfere and break that transcript before it gets to the translation period, so you never actually make the proteins. And that is the last bit of Unit 3. So at this point, you should be able to do the review and feel confident in your answers. And if you need any more information on any of these topics, you can meet with me. Otherwise, I will see you next time.